All right, thank you, Savannah. Thank you, Dr. Williamson. Can everyone hear me okay?
Yep.
Perfect, thank you. My name is John Bleeker, one of the medical oncologists here at Sanford Health in Sioux Falls. I appreciate the opportunity to present a update on a project funded by the DaCCoTA grant entitled Investigating the Utility of Circulating Tumor DNA and Predicting Outcomes Following Definitive Multimodality Therapy for Locally Advanced Esophageal and GE Junction Cancer. Next slide. So esophageal and GE junction cancer is a significant clinical problem. It is the 8th most common cancer diagnosis and the 6th most common cause of cancer death worldwide. You can see in the pie graph on the right the distribution of stages at diagnosis. About 15% of patients are diagnosed with early stage 1 disease, typically cured with surgery alone. On the other end of the spectrum, just over a third of patients have stage 4 disease at diagnosis, widely metastatic, where curative intent therapy is not undertaken. It's really the stage 2, stage 3, that 39 to 40% you see here in the middle, which we call locally advanced. These are patients who have a tumor either isolated to the esophagus but invading deeply into the wall of the esophagus and/or have positive lymph nodes. That is the group that we are really focusing on with this project. Next slide, please. Classically, there are 2 types of esophageal cancer. The first is squamous cell carcinoma. These tumors are typically found in the upper or mid esophagus, and the most common risk factors here are smoking and alcohol usage. Adenocarcinoma is the second subtype, which is typically found in the lower esophagus, with the biggest risk factor here being gastroesophageal reflux disease. As a side note, we have seen a trend over the last number of decades, whereas squamous cell carcinoma was once by far the most common subtype of esophageal adenocarcinoma, we are now trending much more towards adenocarcinoma. Next slide, please. So just to give you some background in how we manage locally advanced esophageal and GE junction cancers. When we started this study in 2020, the standard of care was chemoradiation followed by surgery. So these patients would undergo 5 weeks of daily Monday through Friday radiation along with weekly chemotherapy, followed by a rest period of about a month and then surgical resection. Next slide, please. This is a survival curve from the trial that demonstrated the benefit of this approach called the CROSS trial performed in the Netherlands in 2012. And you can see here, the solid line, chemoradiation plus surgery, increased the median overall survival from approximately 24 months to 49 months, establishing this as a standard of care. Next slide. In the clinical trial setting, the estimated 5-year survival for esophageal and GE junction cancer is about 45 to 50%. As is typically the case, the real-world survival lags behind, and multiple case series would suggest this is about 25 to 30%, indicating we have a long way to go to improve outcomes in this patient population. Next. Somewhat interestingly, during the course of our project, the standard of care for these patients changed. Instead of simply chemoradiation followed by surgery, there was a clinical trial that suggested the use of immunotherapy following surgery improved outcomes. Next slide. This is a survival curve from that trial. You'll note that the endpoint here is different than the first slide that I showed. That was overall survival. This is disease-free survival. We do not yet know what the overall survival data is here because the data is not mature. However, the improvement in disease-free survival, median, from 11 months to 22 months, as demonstrated here, was enough to establish this as a standard of care. That's more of a side note. I don't think it really impacts the questions that we are asking or how we ran our project. We just want to make sure everyone's clear that's the case. Next slide. So one of the questions that hangs over the management of esophageal and GE junction cancer when locally advanced is, what is the role of surgery? We know that approximately 50% of patients with squamous cell carcinoma and 25% of patients with adenocarcinoma obtain a complete pathologic response after chemoradiation. We, of course, can only determine that after doing surgery. But one of the first questions that comes up is, do these patients actually need surgery? I will share with you that in my clinical practice, I have a number of patients who either by patient choice or because of other medical issues do not undergo surgery, and there are certainly a handful of them that have been cured. And so is it possible for us to spare patients what is a morbid procedure if they can be cured regardless? Question number 2, is there a better way to define those who might not need surgery? Is there a way that even in the pre-surgical setting, we can use clinical data to predict who might have a complete response and who may not benefit from surgery, once again, sparing them a morbid procedure? And then the last question, kind of on the flip side, is there a way to predict those who will relapse after standard of care therapy? The thought here is we know that half or more of patients are going to have a relapse, and this is a morbid surgical procedure. If you know a patient is at a high risk of recurrence, is there a scenario where you would say, yes, we've done local therapies, chemoradiation, but because you're at such a high risk of recurrence, we do not want to do surgery. We do not want to put you through this morbid procedure because the expectation is that you will have evidence of recurrence. So this is ways that we could potentially optimize, maximize therapy for these patients. Next slide, please. And so one tool that is coming to the fore that could potentially help us answer this question is circulating tumor DNA, ctDNA. I'm sure many of you are familiar with this concept. This is a cartoon that shows the tumor at the bottom, either by apoptosis, secretion, or necrosis, releases ctDNA into the blood that can then be detected and identified in the plasma. And the current thinking is this may be a very powerful tool to help us answer the kinds of questions that I mentioned on the last slide. Next slide, please. So it has been demonstrated to have utility in predicting residual and recurrent disease. Next. This is data from a large Japanese colon cancer trial. And basically, this took patients who had completed colon cancer therapy, colon cancer surgery, and did circulating tumor DNA testing at multiple endpoints following surgical resection. And here you can see the blue line, if you are ctDNA negative, if we cannot detect ctDNA after surgery, your risk of recurrence at 18 months is less than 10%. However, if we detect ctDNA following surgery, your recurrence risk is over 60%. And so I think the world of colon cancer is probably the most advanced in using this tool. This is starting to enter our research world and the world of clinical practice. And so this is the kind of way that ctDNA can be a helpful tool. And our goal with this project was to investigate whether we can use it in a similar way for esophageal and GE junction cancer. Next slide, please. So here's our study design. Study opened in November 2020. Our goal was to enroll 20 patients over 24 months in the Sioux Falls and Fargo Sanford sites. At the time of diagnosis, the patient would have a biopsy of their tumor and then have blood-based testing, with this sample really trying to identify what is the correlation between ctDNA in the blood and tissue-based gene mutation testing. The patient would then undergo chemoradiation with a blood sample following to see whether that would change the presence or absence of circulating tumor DNA, and then go through surgery with another sample to check if they have cleared their ctDNA at that point, followed by surveillance. Of note, our genomic testing was performed by IPG, and our liquid ctDNA testing was performed by Circulagene. Those were our vendor partners for this trial. Next slide, please. So 3 basic research questions. Number 1, do ctDNA findings correlate well with solid tumor genomic findings? Number 2, does the presence or absence of detectable ctDNA at diagnosis correlate with outcomes? And number 3, does clearance of ctDNA during treatment predict outcomes? Next slide. So we enrolled 12 patients over 24 months. We did not meet our goal. Number of reasons for that we can talk about offline. But of those 12 patients, next. All 12 patients did have pathogenic gene mutations noted on tissue testing. Not a surprise. However, only 5 out of 12 of those patients, 42%, had clinically significant gene mutations noted on ctDNA testing. So less than half of the patients had a correlating finding in their blood as opposed to the tumor. For some reference, in metastatic disease, for most tumor types, this number ranges between 70 and 90%. Next slide. All 12 patients started therapy with chemoradiation. 1 patient passed away before completion of chemoradiation. 10 out of the 12 patients underwent surgery. 1 patient passed away between chemoradiation and surgery. As of last follow-up, 8 of these 12 patients are still with us. 2 of the patients died after surgery in the follow-up period. Next slide. So this is a table. These are all 12 of our patients. And just to go through column by column, the pre-CRT line, the positive indicates that their ctDNA was positive. They had a correlating gene mutation in the plasma when compared to the tumor-based testing. The negatives, of course, mean that they did not. So you can see here 5 out of the 12 pre-chemoradiation indeed had a positive finding. Post-chemoradiation, 1 of the positive patients had passed away, but of the other 4, 3 of the 4 cleared their ctDNA after chemoradiation. Interesting, 1 did not. Post-operative, every patient who was able to undergo a blood test had a negative ctDNA finding. And as mentioned, 12 of the patients are still with us, with overall survival measured in days in the far right column. Next slide. In an effort to better focus these results on the questions being asked, I kind of rearranged these results. The top 5 patients now are those with a positive ctDNA test at diagnosis. And you'll see, as mentioned, that 3 out of the 4 who were able to have tests following chemoradiation reverted from positive to negative and then stayed negative at their post-operative sample. The 7 patients at bottom were negative at diagnosis and remained negative throughout their treatment course when tests were able to be obtained. As mentioned earlier, 3 out of the 5 patients who had a ctDNA positive result at diagnosis passed away, as compared to only 1 of 7 who were ctDNA negative at diagnosis. And you can see on the far right, the median overall survival, 225 days in the group that was ctDNA positive at diagnosis, as compared to 493 days in those who were ctDNA negative. Once again, very small numbers, hard to draw any firm conclusions. Nothing here is statistically significant. But I think the trend that does jump out at me, and I think is hypothesis generating, is the fact that ctDNA positivity at diagnosis may be associated with a poorer prognosis and overall survival. Next slide, please. This is a little bit different way to look at that data in graphic form. I want to consider this as 3 different snapshots. On the far left, entitled pre-treatment plasma, this is at diagnosis. So as we can see here, the top group of 5 dots were those that were ctDNA positive. You can see that once again, 3 out of those 5, 60% had passed away, as compared to only 1 of 7 who were ctDNA negative. The next snapshot, the post-treatment plasma, is patients who have completed chemoradiation. And now you can see that there is only 1 patient who remains ctDNA negative. That patient did pass away. However, 2 of the patients who were ctDNA negative also passed away, meaning there's not a great correlation there with ctDNA clearance and better survival. And then post-surgery plasma, as noted, all 7 patients who were able to undergo post-surgical testing were ctDNA negative. However, even that was not predictive of survival, as 1 of those 7 has passed away. Next slide,
2
please.
minutes left.
Thank you. So conclusions, well timed, Dr. Williamson. In this small cohort, ctDNA correlated with solid tumor genomic results in a minority of patients. My hypothesis there, and this has been backed up by some data, is that in the locally advanced as opposed to metastatic setting, your ctDNA yield will likely be less. You just have less disease to shed that ctDNA. Second conclusion, detectable ctDNA prior to therapy was suggestive of a higher risk of poor outcomes. Once again, hard to say with any certainty based on small numbers, but I think it is hypothesis generating that may be a biomarker we can use prior to treatment to predict outcomes. And finally, clearance of detectable ctDNA did not appear to be predictive of improved outcomes. Hard to say that for certain given the small numbers, but I think that the fact that a number of patients who were able to clear their ctDNA still ended up passing away supports that, at least as a preliminary conclusion. Next slide, please. Thank yous to patients and their families who were willing to participate in this trial, to DaCCoTA for the funding, to IPG and CirculaGene, our clinical testing partners, for 1, doing the test, secondly, for helping us with interpretation and bioinformatics, Dr. Williamson and the Biostats Core, who helped significantly with the statistics here, Dr. Coretta, who lent his expertise in terms of genomics to this project, and Miranda, the glue who held the project together and made sure that we were on track and had our T's crossed and I's dotted. So with that, next slide, have, I believe, a few minutes left for any questions from the group.
All right, you are correct. We have about 5 minutes for questions. So I saw Dr. Ganai's hand go up first. Dr. Ganai, if you want to lower your hand and ask your question.
Sure. Wonderful presentation. I had a question because your outcomes, I know, were designed to look at survival. But in terms of pathologic response, did you notice any thing? I know it's very small numbers. And then to add to that, if you can comment in terms of the role of this, not just for predicting surgery, but for immunotherapy, if you foresee any value.
Yeah, so the first question I will share with you. Great question. We are working on trying to analyze all that data, including recurrence data. You will notice that there are some gaps in some of the blood draw data. And that's because there are a few patients that are not yet due, that had not yet had some of their post-operative draws. So we decided not to include some of that data. I hope to be able to bring that back to this group. And we absolutely could have done pathologic response. We do have that, but we wanted to wait until we got all the post-surgery ctDNA data first. The second question, I think it's a really great question. I will share with you that, I had brought up some of the data from colon cancer. We have multiple open clinical trials here at Stanford right now using ctDNA to guide post-operative therapy. If ctDNA is positive, you intensify therapy. If ctDNA is negative, you de-escalate therapy. And I think my hope and my expectation is that that'll be exactly the same for this particular tumor type. That based on either pre or post treatment ctDNA, it will indeed help guide which patients are going to benefit the most. So it's not a one-size-fits-all approach like it currently is.
Because I currently have 3 octogenarians who are reluctant to have surgery. They could have surgery, but they're frail and it's high risk. And so they are hoping that if they have a clinical pathologic response, that they can do something like a watchful wait where we do endoscopic surveillance every 2 to 3 months. And it would be a really great thing if we could create a trial where we could do this prospectively and also monitor ctDNA, but also determine if there are other therapies that they might benefit for to avoid surgery.
I couldn't agree more. That is exactly the patient for whom this kind of testing is going to be essential. And I just have been a part of some conversations that I mean, every treatment trial that we do right now incorporates ctDNA in some way, in a retrospective type fashion. I am hopeful that through the cooperative group networks in the very near future, the trial that you proposed is going to come to us. Because to make that trial the most meaningful, it's going to have to be done on a large scale basis. But I think it's coming. I really do.
Great. Thanks for that question. I think Dr. Rezvani, you are next.
Hi, Dr. Bleeker. Thank you very much for your nice talk. I was wondering that since you mentioned also colorectal cancer, is there any relation or correlation between ctDNA and age, gender, and a stage of the cancer in this particular type of cancer, or if you've seen in the colorectal cancer?
Thank you for the question. Stage is an absolute yes. It's very interesting because different tumor types shed ctDNA at different rates. And stage also does matter. As mentioned, in metastatic esophageal and GE junction cancers, studies have shown anywhere between 70 to 90% correlation between tumor-based gene testing and ctDNA. Our number here is much lower. In other tumor types, there's also data to suggest that local disease does not have the same rate of ctDNA positivity. I don't think that's a big surprise. So stage does matter. I am less familiar with data, based on gender, age, those kinds of parameters. But stage definitely does matter.
Thank you.
Okay, Dr. Takaku, you're up.
Yes, thank you. And that was very interesting talk. And I have a kind of quick question regarding the detection of the cell, like the ctDNA, because in the blood you can have the DNA from other kind of cells, not necessarily like the tumor cells. And I think how you detect the tumor-specific DNA, and particularly like the subtype specificity or other kind of the cancer specificity, or might be also very important. Do you develop your own like the kind of probes? So sorry, I might have missed the kind of the method that how you detect the ctDNAs.
Yeah, thank you for the question. And it's one of the biggest questions in the field right now. I will share with you that because of, funding concerns, we're not doing our own testing here. Basically, what we did is use clinically available tumor and ctDNA testing, and then compared those to see how the correlation was. Whether genes that were identified in the tumor were identified in ctDNA, that was considered positive. If they were not, that was considered negative. There are, and the trial that I showed you in the colorectal space utilizes a bespoke probe. So you send the tumor sample in, they create a unique probe for that individual patient's cancer. And I think there's a lot of reasons to think that is probably going to be the gold standard. We don't know that yet. There are other trials that do not use a bespoke probe. They use a probe that investigates for 8, 10, 12, 20 different gene mutations that are common in a particular cancer type. So your question is a very good one, and I think the field is still trying to sort out what the right way to do this is. I will say, just because of our resources, we were not able to do that kind of next level patient specific testing.
Thank you so much.
Yeah.
All right. Well, thank you so much, Dr. Bleeker, for your presentation. Next up, we have Dr. Kevin Francis.
Thank you, Mark. Thanks for the opportunity to present my work today, and organizers. So I'm going to tell a story today from our feasibility project that links three different perspectives related to basic biology and cancer biology. So I'll start with my acknowledgements. First, thanking the DaCCoTA program for supporting the project. I'm going to summarize today. I think we have some really interesting findings that we're trying to disseminate currently. I'd also like to thank the individuals who performed the work. You'll see primarily Jasmine Yeager, who's a postdoc in my lab, who was supported by DaCCoTA, as well as a few other members of my laboratory. I'd like to thank Drs. Vermeer and Spanos here at Sanford Research for collaborating on this work. This is my first foray into cancer research, and so I greatly appreciate their support on this. I also realized I forgot to acknowledge the support from the flow cytometry core here at Sanford Research. So apologize to Jared and Pilar on that. So next slide, please. Sorry, go ahead. Next slide. I forgot. So I have no conflicts of interest. Next slide. So our disease model of interest for this study was head and neck cancer squamous cell carcinoma. So over 50,000 new cases of head and neck carcinoma are identified in the US yearly. About 75% of these are HPV negative and caused predominantly by tobacco use, while about 25% are result from HPV infection. Treatment involves combination of surgery, radiation, chemotherapies, and new immunotherapies, but these have a varying degree of success. HPV positive cancers usually have a improved treatment response and survival compared to HPV negatives, for example, potentially related to immunogenicity differences between the two. So for this project, we're really building upon a 2018 publication that I'm highlighting on the left-hand side there in Nature Communications from Vermeer and Spanos labs, along with some other labs where they demonstrated that small extracellular vesicles that are called exosomes are specific and important contributors to tumor innervation, and this is associated with disease severity. And we know tumor innervation is related to higher innervation of solid tumors is related to worse prognosis and in patients. And so they identified that exosomes contribute to innervation, and also that exosomes derived from head and neck models actually increase tumor innervation, and that's highlighted at the bottom in that fluorescent imaging where they're staining for using immunostaining to look for proteins associated with innervation within the tumor itself. Next slide, please. So just a quick reminder of what exosomes and extracellular vesicles are. So exosomes are small extracellular vesicles that are approximately 100 nanometers in size that can function as a cargo for transport of different molecules like proteins, microRNAs, lipids between cells and tissues. They circulate within the periphery and have been implicated within a large number of diseases, cardiovascular and cancer in particular. So next slide. And so the final piece for this kind of project puzzle where we're linking head and neck cancer, exosomes, and lipid metabolism is actually cholesterol. So my lab's principal focus historically has actually been cholesterol metabolism and looking at how changes in cholesterol synthesis and turnover lead to or contribute to a variety of diseases. Within the realm of cancer, a lot of studies have correlated high levels of cholesterol and high rates of cholesterol metabolism are associated with cancer progression and cancer incidence. However, the mechanisms whereby cholesterol regulates cancer, contributes to cancer, are kind of varied, and it ranges from regulation of signaling pathways, stabilization of receptors like receptor tyrosine kinase receptors within the membrane, and downstream signaling, as well as just overall changes in cell metabolism. And then my graph on the right, which is supposed to be a heat map, but that didn't turn out very well, is supposed to be showing that there's a correlation between the expression of cholesterol synthesis genes and in this case, this paper was showing melanoma survival. So higher expression of cholesterol synthesis genes are associated with poor prognosis in melanoma. Next slide, please. Exosomes and extracellular vesicles are as carrier molecules are extremely lipid rich. Within EVs, there have been a number of lipidomic studies done to analyze the lipid profile of EVs, and they found that EVs constitute approximately 45% or 40% of the cholesterol is within these EVs of the total EV lipid profile. So given the high amount of cholesterol that's present within these EVs and the link of cholesterol to cancer, as well as the link of EVs to cancer. So next slide, please. Yep. So our hypothesis was that inhibition of cholesterol synthesis would actually impair EV production and release, and this could potentially have benefits impacting head and neck cancer patient outcomes. And I'll talk about what we're trying to do with patient samples towards the end of the talk. Next slide, please. So to resolve the impact of cholesterol metabolism on EV and exosome production and function, we utilize a number of different small molecules which target various steps within the cholesterol synthetic pathway. You don't need to know anything about any of these structures other than we are targeting different enzymes to see if we're getting the same effect, and it's not just a single inhibition of a single protein. You might recognize some of these molecules like statins that are inhibiting HMG-CR upstream of the pathway, and we're also utilizing some downstream small molecules to inhibit other enzymes within the pathway to try and correlate findings across the pathway. So next slide. So to start, I wanted to highlight the fact that different cell types can exhibit a varied biochemical response to cholesterol synthesis inhibition. So here I'm showing GCMS data where we are treating 3 different cell types, human cell types, an embryo cell line derived from human embryonic kidney, the HEK293T, human keratinocytes, HCATs, and a human immortalized human microglia, HMC3. So in the top right graph, you can see there are differences in the basal amount of cholesterol that's present within these cells, suggesting that these different cell types may be are exhibiting changes and differences in synthesis or turnover or usage of cholesterol that we aren't clear. And in the bottom graphs, when we treat these cells with our small molecule inhibitors, we can see a reduction in cholesterol levels where the bars are generally lower than the vehicle baseline. But you can also see that there's a varied degree of inhibition across these cell lines, as well as accumulation of a number of these other sterile precursors, molecular precursors of cholesterol that are highlighted in the different colors of the bar, which are structurally distinct from cholesterol. So next slide, please. In order to quantify the impact of cholesterol on EV and exosome number and size, we first isolate EVs using a series of centrifugation and purification steps, and then we take the purified EVs or exosomes and image them on a nanocyte imaging system that we have here at Stanford Research. We analyze them in suspension. We can actually visually see the EVs kind of flowing through the channel, and we can use these to quantify the size of the EVs and also and normalize the number of EVs that we're quantifying to our cell count, to a cell count that we from the original starting population of cells. Next slide. So in quantifying the impact of cholesterol inhibitors on EV production, we actually found the exact opposite of our hypothesis. So we had assumed that by reducing cholesterol levels, cholesterol being so highly expressed in EVs, we would see less EVs. But we actually found the opposite. Where in the top bar graphs, we found that EV secretion are actually dramatically increased following cholesterol synthesis inhibition, though there are some differential impacts across these cell lines depending on the small molecule utilized that at this point we don't understand why that would be, because the cholesterol is reduced in all of them. In the bottom graphs on the left-hand side, we don't see a dramatic shift in vesicle size across the cell types. That was a concern of ours. We thought maybe the vesicles could be smaller, but we don't see a change in size. And in the line graphs on the right, we see a really high correlation across all three cell lines between cholesterol levels and EV production. So more cholesterol actually equals less EVs. I would also note that all of the drugs that we're using here, we performed dose responses and used a concentration that gave us a maximal biochemical response without inducing cell death in these cell lines. So next slide, please. So determine if cholesterol inhibited EVs have a distinct molecular profile from controls. We utilized a kit from Miltenyi Biotech called MaxPlex, which utilizes a panel of antibodies which recognize surface proteins that are commonly found within pools of extracellular vesicles. So for these assays, the EVs are collected as we did before, and then we analyze them for expression of these markers by flow cytometry, and they're sorted into these pools of extracellular vesicles based on the marker expression. So next slide. So one interesting thing we see, for the most part, it's hard to see based on the graph, but for the heat map, the three main EV markers that most labs utilize, CD9, CD63, CD81 on the left heat map, and we see very subtle changes in these major EV markers independent of these treatments. However, we did identify within this panel of markers, we identify some changes in some other proteins which may be really relevant to immune activation and tumorigenesis in head and neck cancer and maybe potentially other cancer models. So for example, we see increased expression of CD56 and CD3, which are both T cell receptors. We see increased expression of ROR1, which is associated with neurite outgrowth, as well as increased integrin expression related to cell adhesion. So we're still trying to validate these findings with some more focused assays, but we also interestingly see some differences again across these inhibitors that we're using, and that we're still trying to figure out why one inhibitor versus another may be impacting, maybe giving a defined molecular profile versus another. So next slide, please.
2 minutes remaining.
Okay, thanks. So we don't know mechanistically why cholesterol may be impacting EVs this way, but it may be related to a general cell response related to intracellular trafficking, where we see increased expression of these endosomal markers, RAB5 and RAB7. Next slide, please. And within the context of EV, we see, from a GCMS perspective, we see a dramatic differences in the response of cell lines derived from HPV negative patients outlined in blue versus HPV positive patients outlined in red, where there's a very different response to these inhibitors across these cell lines, which may be related to genetics or the cell line of origin. Next slide, please. When we treat our head and neck cancer cell lines with these inhibitors, we also see a dramatic increase in EV production, both in FBS conditions, potentially related to non-cholesterol impacts, as well as in these lipid-free conditions on the right-hand side, where we see a huge increase, particularly in HPV negative cell lines. Next slide. We've been trying to look at a number of different parameters for these cell lines and cholesterol impacts. And we do find one parameter I'm showing is HPV negative cells are particularly sensitive to these cholesterol synthesis inhibitors, where we see increased cell death, even at fairly low doses of our cholesterol synthetic inhibitors. And we're still trying to figure out why that might be. Next slide. So in summary, we see changes in EV production in both control and head and neck cancer patient cell lines. We're looking at the function of these EVs. Can they be taken up? Do they impact things like neurite outgrowth, tumor innervation? We're currently looking at patient plasma from head and neck cancer patients that we received from Dr. Spanos, and also interested in other cancer types. Is this common across a number of different cancer models? So, and next slide will be my acknowledgements, and I'll take questions if I have time.
All right, excellent. Thank you for that talk. Yes, we have 5 minutes for questions.
Dr. Francis, very nice talk. I just wondering that, is it possible one of the mechanism behind this elevation of the exosome release is that this molecule basically auto-generate a heat shock response, stress the cells, and then based on this question, whether examining the level of the heat shock protein inside the cells or in the exosome would be worth it to look at as a underlying mechanism, one of the underlying mechanisms.
Yeah, no, that's a really good idea. I actually had not thought of heat shock response. We're looking at currently looking at ER stress, which I think is associated with these inhibitors. Heat shock response we have not looked at. It's really with being new to the exosome field, getting material enough to look at defined pathways can be pretty challenging. So, but heat shock, yeah, that's a great idea. Yeah, I'll look that up, Khosrow. Thank you.
Thank you.
Hey, Kevin, just a quick question, and maybe you went over this already. When you see that increase in the number of exosomes, do you also see an increase in other vesicles that, you know, when Jasmine does the differential ultracentrifugation, are there more microvesicles or more apoptotic bodies as well? So is it a generalized phenomenon or limited to exosomes specifically?
So at this point, I don't know. I have a -80 full of these different phase separations from these centrifugation steps that is on Jasmine's to-do list. But so we don't know yet, Pei. That's a good question.
One other question, I can't remember if we have the information, I think we do, on all those head and neck cell lines, whether they're male or female. It might be interesting to go through and see if there's a sex difference as well.
Sure. Yeah, I don't know if we ever talked about that. That's a good idea though. Yeah, I should go back and go look. I don't know off the top of my head which cell line is which sex. Yeah.
Are there any other questions? Okay.
Thank you. Appreciate it.
All right. Well, thanks again for that talk. Great. So next up is Dr. Soojung Kim. So when you're ready to go, you can get started.
Thank you. Thank you for having me today. My name is Soojung Kim, and then I would like to present a study that was supported by the DaCCoTA Feasibility Award, and then some of the updates afterwards. So next slide, please. I'd like to start with the acknowledgement and express our gratitude on behalf of our team to DaCCoTA for the pilot grant, which was supported this study and other efforts moving forward. Next slide, please. So this study is really about communicating the risks about radon, and radon is the second leading cause of lung cancer after smoking and cause 21,000 deaths per year in the US only. And 1 in 15 homes has radon levels higher than the EPA recommends as a safe level of radon, indoor radon levels. Next slide, please. Before this DaCCoTA study, we had a small internal grant that made the pilot intervention study available. With that study, we developed a novel mobile app to educate the public about radon's risk, and we tested its feasibility and effectiveness on increasing radon knowledge and radon testing. And we published it in BMC Public Health, where the overall conclusion is that the radon app is working. It's a promising tool for radon education. So with that app and this pilot study, we applied for the DaCCoTA grant. Next slide, please. So the goal of this project with DaCCoTA feasibility grant was to conduct a randomized control trial based on that pilot study, and then compare the performance of the radon app versus the same information delivered via printed brochure, which is a traditional form of communication in terms of increasing radon knowledge, making more positive attitudes toward radon testing and actual testing behaviors. Next slide, please. So our first hypothesis was that there would be more positive changes or greater positive changes among the radon app users compared to the brochure recipients in terms of their knowledge levels, their attitudes, self-efficacy, and response efficacy. Self-efficacy in this context is about an individual's beliefs or perceptions that they can actually conduct radon testing by themselves. And response efficacy in this context means that whether individuals believe that the radon testing is actually effective in preventing lung cancer to develop. The next slide, please. The second hypothesis that we had was the incidence rate, that incidence of participants' radon test kit request and the utilization request would be higher among the radon app users compared to the brochure recipients. Next slide, please. So we conducted a 2-arm pre-test, post-test randomized control trial. We recruited participants. They were randomly assigned to one of the 2 conditions, so either radon app or the brochure condition, and they completed the pre-exposure survey, which was the baseline data, and then exposed to one of the two conditions, either radon app or brochure. And then they came back 3 months later to complete the post-exposure survey so that we were able to compare pre and post test data. Next slide, please. Participants were 148 undergraduate students from introductory comm classes here at UND. All of them were 18 years old or older, and then they were smartphone owners. And then if they themselves or their parents have tested radon for their house in the past 2 years, they were excluded from participating in the study. Next slide, please. So here is the little procedure kind of a figure. So the participants came to the research lab here on campus at UND, and they completed the pre-exposure survey. And again, that was the baseline data. And then at that point, they were randomly assigned to one of the two conditions, so either radon app or brochure. For the radon app condition, they installed the app on their phones after completing the pre-exposure survey, and they were asked to use the app for 3 months, just as they would for normally other apps on their phones. And they were asked to access to the in-app content that was available for that 3 months period. And then they were given an opportunity to order free radon test kits at any time using that app. On the contrary, brochure condition, they received the 3 EPA created brochures for 3 months and once a month for the 3 months period. And then included in each email or included in each mailing, there was a free radon test kit order form. They can fill out its pre-addressed, pre-paid form, so they can just drop it in the mailbox. And after that 3 months period, they were invited to come to the lab again to complete the post-exposure survey. And some of the monetary compensations were given to both pre and post-exposure survey. Next slide, please. And these were the measurement scales that we used. All of them were used in previous studies and then tested as reliable and valid tools to measure these variables of interest. Next slide, please. This is a concert form for our randomized control trial. So we recruited 148 participants who for the eligibility check first. 10 were excluded because they had done radon testing previously. And so 138 participants were randomized, 70 assigned to the radon app, 68 assigned to the brochure condition. After that 3 months period, we lost 13 from the radon app condition and 18 from the brochure condition. So overall, we had 57 left for the app condition to analyze their data and 50 for the brochure condition. Next slide, please. Our first hypothesis was about the greater positive changes among app users compared to the brochure recipients in terms of their knowledge, attitudes, and to efficacy outcomes. We've seen a very interesting results showing that indeed radon app users showed a greater increase from pre to post test survey in terms of their radon knowledge. So they learned better among those radon app users compared to the brochure recipients, and their self-efficacy level increased more significantly greater than the brochure recipients when we are comparing pre and post test survey data. Next slide, please. Then we expected that there would be higher incidence of app users' requests for the free radon test kits and their utilization of those test kits. We do see a greater or rates or incidence of participants among the app condition requesting the free radon test kits. So it was about 40% of the radon app participants ordered the free radon test kits, compared to about 13% of the brochure recipients ordered the free radon test kits. But utilization rates were very small in both conditions, that only 2 out of that 29 app condition participants who requested free radon test kit, and 2 out of 10 brochure condition participants who ordered the free radon test kit actually used them. So those numbers were too small to compare. So that was one of the things that we didn't expect to see, but that was actually an interesting finding ultimately. Next slide, please. Some of the limitations that we wanted to mention is that we approximately lost 23% of the participants because it was a pre and post survey design. It was slightly bigger than the typical behavioral science research when they are doing the longitudinal study. But then that was something to mention, that we lost about a quarter of the participants. And then it was a university student sample. So there is indeed a generalizability issue there, because many of them, if not all, are not homeowners. They lived in a rented apartment or university housing. So, but then it was still meaningful because based on this publication and support from the DaCCoTA, we were able to receive an external grant, which led us to actually do the replication study of this with the homeowner population. And it was done in a lab setting where students had to come to the research lab on campus for both times, pre and post exposure, which is very artificial setting. It has some external validity issues for sure with that setting. Next slide, please. With that in mind, though, we concluded that the Radon app is a more effective tool for radon education and then encouraging people to radon testing than the printed brochures. But then some of the areas that we wanted to focus after the study was that how can we actually increase the radon test kit usage rate? How can we actually motivate them to act on it when they have the free radon test kits? And also, since it was an artificial lab setting, and then so when we were to disseminate this app, how can we actually promote this app for individuals to download in the first place? So that was beyond the scope of the this study, but then that was a question that we had as an important next step for our group. Next slide, please. So these are kind of summaries of the main findings that we did. I wanted to express our gratitude on behalf of our team for DaCCoTA and the supports that we have throughout this grant period. And then again, this funding and the publication that we were able to publish led to a different external grants opportunity where we are tackling those generalizability issues and different ways to testing, radon testing, so that we can increase the test kit usage rates. I'd be happy to take any questions if you have any. Thank you.
I actually have a question for you, Dr. Kim. So now that you're going to be eventually looking into how the app is utilized by homeowners, do you anticipate possibly there being any difference with age and utilizing the app versus the paper brochures, like in a older participant audience?
Yes, that's a very good question. We understand that the younger generation, university student population, traditional students are younger, they're more tech savvy, they're more familiar with the digital technology. interestingly, when we did the replication study as a follow-up study of this DaCCoTA funded study, when we were testing the same exact design with the homeowners population here in Grand Forks, app was still a very effective tool to order the test kits because it was just so easy. You just click through or just touch through some of the steps, and then it will be delivered to your home like in a few days. However, the usage rates were significantly, I believe, higher in the brochure condition than the app, which was unexpected than from our original expectation. And we kind of thought that that's related to that age group. Obviously, the homeowner population was much older. I believe the average age was 35, 37-ish, which is almost 10 years or even 15 years older than the university population average age. And then perhaps brochure and then have that physical reminder help them to remind them of testing using that test kits that they received. So yes, I think that Radon app might work better for the younger users. And I do believe as a communication researcher, you cannot really hit all the demographics with just one single communication platform. So if we were to actually disseminate this app to the general public, we would probably have to do some media planning, that Radon app more targeted to the younger generation, and some other means, perhaps that would be the brochures, other more physical forms of communication to reach older generation.
All right, we have a question in the Q&A. The question is, have you come up with any methods to promote the actual use of kits versus just ordering the kits?
We haven't done that question. However, what we found from this study and a couple other follow-up studies that we did is that the test kit usage rates are not so much of a communication problem. Radon knowledge was a communication problem, or the awareness of that issue was a communication problem, and that was effectively solved by the app, we believed. However, test kit usage is more like the test kit itself that we use for a free radon test kits are short-term test kits with the charcoals. So participants have to place that in their basement for at least 48 hours. They have to fill out a form physically and then mail it back to the lab for analysis. Then they will get the results in a week or so. They have to interpret the findings. So there were so many different hurdles for individuals to have to go through when they're using the short-term test kits. So we thought, well, that's not really so much about reminding people to use it, but then the test kit itself has to be more simple. So one of the follow-up studies and project that we've been doing so far is to bring in digital radon test kits, which you don't have to do those steps. You just simply reset the digital test kit. It's about the cell phone size, and then put it in your basement and then leave it there as long as you want. The longer you put it there at your home, the more accurate the numbers are. And then your radon level number will pop up on the screen right away. You don't have to send it back to the lab. You have to interpret it, wait. And sometimes when you are sending the test kits to the lab too long, like over a week later, I believe, the numbers are inaccurate. And then sometimes they have errors because individuals didn't follow the instructions carefully. So even if you send them to the lab, you won't get the numbers. So there were so many human errors that will be involved in that short-term test kit setting. So I think, and then we have the digital radon test kits made available through our Grand Forks Public Library. It's been received very well by the public, at least the Grand Forks population. So I think that might be one of the promising ways to solve that usage problem moving forward. So we'll see.
All right, thank you so much, Dr. Kim, for your talk. Next up, I believe we have, Dr. Takaku on behalf of, I think, Dr. Wang's talk or project. All right, when you're
Yes,
ready, you can start.
Thank you so much. So actually, I was a co-investigator, and because Dr. Wang left here, so I'm going to share the result. First of all, thank you so much for giving us this opportunity to share our result, and also selecting us for the pilot grant. And next slide, please. And I have to first emphasize that this is like the teamwork and group study, and we are getting a lot of support, and particularly from Sanford Group and Dr. Ganai and Ms. Emka Gary. So without, and also local patients, so without those help, so we couldn't do any studies that we are doing now. And we have also very talented grad student, Nazim and Sakunsa, and they are actually the one who is collecting the data and analyzing the data. Next slide, please. Oh, there was some error. Okay, so anyway, so what we have been doing is trying to develop some new technology or technique or method to for the cancer diagnosis. And obviously, early detection of the cancer patient or cancer is very key for the survival, patient survival. And this kind of COVID-19 pandemic also affect the screening, and many patients are actually facing some delays of the screening. So that might go to the can impact on the kind of detection at the early stage. But so we are kind of trying to develop something new that can be easily test the cancer patient. Next slide, please. Oh, and so what we are trying to use is there was a very nice talk, circulating tumor DNA talk from earlier, but so we are doing something similar, but kind of trying to use a slightly lazy way. So we are trying to analyze all DNA that you can find in blood. So cell-free DNA is a DNA you can find in the bloodstream. And it has been known that this cell-free DNA is known more than 50 years ago, and we know that in the healthy, like the donor, so you can see that their DNAs, those are coming from mostly immune cells, so we can see some signs of the immune cells. And but in the case of the patient, if you take blood from a cancer patient, we can actually see some signs of the DNAs that are coming from those tumor cells. Next slide, please. And at early the stage and in the current clinical setting, many people are focusing on the cancer-specific mutations, but that has some limitations to detect some specific cancer type or stage or other type of informations. And now we know not only like the DNA mutations, we can detect some partially conserved epigenetic information that can be DNA methylation or chromatin structure. Next slide, please. So I have to first probably explain what chromatin and why we are excited about the chromatin structure preservation in this cell-free DNA. So when we think about the cells and any different types of cells, they have only one set of genome. But how the cells can change this cell property is that simply changing this epigenetic information that is essentially chromatin structure. Next slide, please. So this is a very simple scheme and how the chromatin structure affect the gene expression or cell properties. So the fundamental unit of the chromatin is this small b, so it's called the nucleosome. And you can detect the nucleosome everywhere in the genome. And when the nucleosome is physically compact or condensed, those regions tends to be gene expression tends to be silenced or inactive. However, when the chromatin physically become open, and now the transcription factors or polymers can access to those DNA. So that's why the gene expression become active. So those are the kind of basic fundamental mechanism of this epigenome that we can see in the our human genome. Next slide, please. And very surprisingly, so by just simply looking at this chromatin structure, we can detect or predict any types of cells. So in this slide, what we are seeing is signals from many different types of cancers. And when you see the signals, so this signals means the open chromatin lesions or active chromatin lesion. Whenever you see the signals, those lesions are active. And some regions you can see like the very common across different types of cancers, but some regions are very specific to some specific type of cancer. So that's why if you can detect the chromatin structure, that can be used to predict the cell origin or cancer subtype or cancer type. So that's kind of the reason why we are so excited that we can now detect the chromatin structure even in this cell-free DNA. Next slide, please. And this is not kind of our own kind of novel idea. So some of the groups have been already showing some of the aspect, and there is some kind of proof of concept study. So by looking at some epigenome that in cell-free DNA, you can predict some of the cancer cells or cancer types and or some kind of disease tissues. And that is not necessarily only limited to cancer. Then some other human disease can be also detectable by looking at cell-free DNA. Next slide, please. But so the kind of the question in the field is where do they really coming from, and has there any specific chromatin regions or structure that you can see in the specific cancer type, or how much can we use those data to predict or accurately predict the cancer patient, or potentially can we guide a cancer treatment or patient treatment by looking at this cell-free DNA? So those are the things that we are trying to address. Next slide, please. So, but because this is very new to us, and I believe this is new to the our school and could be like in the state. So what we started is just simply starting from the very simple in vitro model. So this is actually like we got the pilot grant from EPSCO and the ACES before we got this pilot grant. So we first kind of collected the very fundamental information. But we essentially did a very, try to recapitulate the cell-free DNA in vitro. So we culture cells and take supernatant and look at the DNA fragment and do the sequencing. And what we saw immediately is that we see some signs of the chromatin structure or nucleosome. So next slide, please. And that's also true in the computational DNA analysis. We can see one nucleosome, or two nucleosomes, or three nucleosomes. So it's kind of chromatin structure seems to be actually preserved in our in vitro cell culture model. Next slide, please. And when we look at the data, so this slide is showing the one genomic location right on the FOXA1 gene. So this gene is known to be highly expressed in luminal breast cancer. And the top panel is the data from ATAC-seq. So that is signals from open chromatin lesion, as I mentioned. So this lesion is active in this in vitro T4-T7D breast cancer cells. So this is kind of reference that we can use as a kind of marker. And the second and third, like the slide, like the panels are coming from this our in vitro cell-free DNA. So we see some enrichment, higher enrichment around this the gene promoter region. So that kind of the signs of like some DNAs might be, cell-free DNA might be coming from those active open chromatin region. Next slide, please. So, and when we zoom out, so we see some kind of conservation. Also, those cell-free DNA data look very noisy. So you see a kind of not like striking enrichment, but some kind of peaks, like the enrichment seems to be conserved between the active chromatin region signals shown in the top panel versus like the second and third cell-free DNA in vitro data. Next slide, please. And of course, that was not only one genomic location. So we confirmed this higher enrichment from active chromatin region in the genome-wide manner. So we can see a very nice enrichment at the center of this active chromatin region. And we also have the kind of control, like the randomly selected genomic regions, and that we didn't see source enrichment. Next slide, please. And then when we went to the patient samples, and previously we collected data from breast cancer patient in this pilot grant from DaCCoTA program, so we could collect the data from the pancreatic cancer patient. And so again, so we see the very nice signs of the nucleosome structure in this data. Next slide, please. And that we confirm this in the computationally, so we see these signs of the nucleosome fragment in these patient samples. Next slide, please. And so this is another kind of example of the data. And in this case, we are seeing the breast cancer and healthy donor derived cell-free DNA. So that is the third panel and the fourth panel. So we see some differences in terms of the enrichment, particularly at this loci, so slightly kind of upstream from the TFF2. So TFF2 is also important breast cancer marker. And then so we kind of confirm the same phenomena, the higher enrichment of the active chromatin region from both from in vitro cell-free DNA and the patient derived cell-free DNA. Next slide, please. And then we wanted to confirm if we can see, or if we wanted to see if we can detect some differences between the breast cancer derived like the cell-free DNA versus healthy donor derived cell-free DNA. And we see actually higher enrichment at this breast cancer specific open chromatin or active chromatin regions compared to healthy donors. And that was statistically significant. So this is kind of the left panel showing the average and right panel is showing some quantitative analysis. Next slide, please. And then, so that was not only specific to breast cancer. When we collect the pancreatic cancer patient data, we see actually higher enrichment at pancreatic cancer specific active chromatin region. Next slide, please. And we also wanted to know if we can detect some differences, cancer-specific differences. And when we look at the pancreatic-specific open chromatin or active chromatin regions, we see higher enrichment from pancreatic cancer-derived cell-free DNA compared to the cell-free DNA coming from breast cancer patient. Next slide, please. So now kind of we got some idea of where to look and how different they are potentially. But because of the limitation of the number of samples, we went to the public data and we downloaded the data from 67 breast cancer patients, 67 healthy donor patients. And this time, so we used artificial intelligence, like the based technique or machine learning based technique to do some prediction analysis that kind of considering the clinical setting. And we got a very high prediction accuracy, at least from this breast cancer patient data cohort. Next slide, please. And what is special with this machine learning technique is that unlike other deep learning or neural network algorithms, it's very easy to extract the very important region, chromatin region that can be used for the prediction. So now we are kind of trying to further evaluate those differences and just to see our model can apply to our data analysis. Next slide, please.
2 minutes remaining.
Yep, thank you so much for your kind attention. And as I mentioned earlier, so this is a teamwork study, and Xusheng Wang, Dr. Xusheng Wang is the PI of this pilot grant. And so I'd like to thank again for the patient and a member from Stanford and our laboratories. Thank you very much.
All right, we'll now have time for questions. Okay, I'm not seeing any questions in the chat or the Q&A, so Unless anyone else has wants to get on the speaker to ask a question or type in a question, we'll start transitioning to the last awardee update of the morning. All right, and that'll be Dr. Huber or Huber. Sorry if I got that wrong.
No problem. Good morning. Thank you. I'm Victor Huber, and I'll be talking to you today about some of the work we did on a SARS-CoV-2 pilot project, looking at antibodies against SARS-CoV-2 and biomarkers in cancer patients, with this one focusing on the difference between the pediatric population and the adult population. And I'm at the University of South Dakota. Next slide, please. So the talk will go through some general features of SARS-CoV-2 to remind everybody about this virus, go through what we saw with the immune response against SARS-CoV-2, and then discuss a little bit about the biomarkers that we saw in a couple of the patients that were a bit notable. Next slide. So moving through the general features of SARS-CoV-2 and COVID-19, next slide. Looking at the virus, this is a virus that binds, it's a coronavirus, binds to its receptor on the surface of the cell. The receptor specifically is the receptor, the angiotensin converting enzyme 2 receptor. It's recognized by the spike protein of SARS-CoV-2. And that virus then gets taken up. Its nucleic acids are released into the cytoplasm, where because this virus is a positive sense RNA genome, it has the ability to be translated directly into proteins that can make more copies of the virus and express some of the structural proteins. These structural proteins are then expressed within a unique compartment known as the endoplasmic reticulum Golgi intermediate compartment, or the ERGIC. And that is where the virus then forms a new virion that can then be released through exocytosis and escape the cell. One of the sort of interesting aspects of the SARS-CoV-2 infection cycle is that it does kind of hide out a bit inside the cell without having any proteins expressed at the surface that sort of identify the cell being infected. And it's one of the things that may be important in understanding the humoral immune response that develops against this virus. Next slide, please. So the symptoms of the coronavirus, these are the things we've come to know over the last few years. Typical to many other respiratory viruses: cough, shortness of breath, difficulty breathing, fever and chills, muscle and body aches, vomiting and diarrhea, and then pretty characteristic of SARS-CoV-2 is that new loss of taste or smell, although that symptom has come and gone throughout the course of the pandemic, at times being observed and then disappearing and then coming back. So, but we now have pretty good tests that tell us patients have SARS-CoV-2. Next slide. So this isn't the only coronavirus. There are 7 types that have been known to infect humans. The first 4 fall into the endemic coronaviruses. These cause about 5 to 25% of the common colds. These include the OC43, NL63, 229E, and HKU1 types of the virus. And these ones, again, cause really the common cold, part of that sort of seasonal respiratory virus sort of panel that we see. And the symptoms and sort of illnesses don't usually get to be too severe. Moving on to the other 3 types, the original SARS-CoV, which was seen in 2003 to 2004 in about 24 countries, caused about 8,100 cases with an 8 to 15% mortality rate. It spread, but not quite as well as we know SARS-CoV-2 did. MERS-CoV is the Middle East Respiratory Syndrome coronavirus. It's been sort of moving around since 2012. Most of the cases are in Saudi Arabia, 2,500 cases, 38% mortality, and it primarily moves from animal to human. So it's not one that's established at human to human transmission. at least not yet. And then, of course, SARS-CoV-2, the one that we're most familiar with, started spreading in 2019, and pandemic was declared in 2020. To date, it's caused 692 million cases and 6.9 million deaths worldwide. In the U.S., 107 million cases with 1.7 million deaths. The end of the public health emergency was declared a few months ago, but that does not mean that the virus is gone or that we are not concerned with SARS-CoV-2 and coronavirus infections. Next slide, please. So this study was started with myself, Dr. Khosrow Rezvani, also at the University of South Dakota, and then clinical collaborators, Dr. Santiago Lopez, Dr. Fernando Bularudis, and Dr. Kaylin Wagner. And this is a team that's been working on these two projects, one looking at adult immune responses and one looking at pediatric responses. Next slide, please. So in order to look at the antibody responses we're interested in measuring, we had to express proteins of the coronaviruses. To do this, the best way that we had at the beginning of the pandemic was to express the receptor binding domain. We acquired plasmids from Florian Kramer's group at Mount Sinai and expressed those in 293 T cells, collected the samples, pelleted the cells, lysed them to release the protein, and then went through a number of column and filtration methods to purify the proteins. This work was done by Ben Kalesa and Matthew Schmitz in my lab, and we moved forward with looking at the antibodies in patient samples, detecting different antibody isotypes, as I'll present on the next slide. So if you look at the results here, this shows the titers of antibody levels in patient samples. Each individual dot represents an individual patient sample, with three groups represented along the x-axis, red being no COVID, green squares being cancer or non-cancer COVID, and then the purple triangles being cancer COVID. On the y-axis, we show the endpoint titer. This is the last dilution where we see a color change in our ELISA assay. Top three panels are the adult responses, with bottom three being the pediatric population. And when you look at the antibody results, you can see that there was really no difference between non-cancer and cancer COVID patients in the IgM, IgG, or IgG1 antibody responses. When you go to the pediatric population, the same is true. IgM, IgG, and IgG1 levels are pretty similar between the two groups. One thing I will point out that makes this data set pretty unique and pretty interesting, the adult patient samples were from patients early in the pandemic before we had vaccines, and many of these were their first exposure to SARS-CoV-2. So we had a pretty unique data set that we could use. And similar to what other people saw during this phase of the pandemic, there was not a uniformly strong antibody response against this virus, and that's kind of what we're seeing in the adult patient samples. The pediatric population came later, and we included vaccine recipients, so that's why we see a little bit more of a uniform antibody response within that group. Next slide, please. To detect some functionality to our antibodies, we did a neutralization assay using fluorescent pseudoviruses that we acquired from Adam Hoppe at South Dakota State University. And this assay was done to show the ability of the antibodies to neutralize the virus infection. Next slide. And again, looking at the antibody responses, we show an endpoint titer as our neutralization titer. And you can see the three groups: no cancer COVID, and then cancer COVID for the adult and the pediatric populations, with the titers on the y-axis. And you can see a more uniform response in the pediatric population, maybe a little bit higher average titer, and then the adult serum was more spread out. What was surprising for me was the fact that the neutralization assay performed a little bit better than the ELISA. I kind of expected the ELISA would have higher titers because that just relies on binding of the antibody to the epitopes. but we also were restricting our binding due to the receptor binding domain and not the entire spike protein. So that may have played a role in the differences between ELISA and neutralization. But we did see neutralization titers in a lot of our samples that we collected, with no difference between the two patient populations. Next slide. Aim 2 of this proposal was looking at biomarkers, and this work was done by Dr. Rezvani here at USD. And the main point was to identify proteins in SARS-CoV-2 positive patients, SARS-CoV-2 negative patients, and look at some of the shared proteins and validate these by ELISA, flow cytometry, and Western blotting. And if we move to the next slide, we'll see that Dr. Rezvani was able to show apolipoprotein E and hepatocyte growth factor had exclusive elevation in 2 breast cancer patients with COVID-19. These make a little bit of sense because ApoE plays a role in the viral infection process, and HGF plays a role in some of the neutrophil responses against this virus. Next slide, please. So in summary, we showed that natural SARS-CoV-2 infection didn't induce a strong antibody-mediated immune response. Again, this is something that was similar to what others were seeing at around the same time. And we saw this with both ELISAs and neutralizing antibody titers. In both the adult and the pediatric populations, the antibody responses were similar between the cancer and non-cancer parts of the patients in the study. And ApoE and HGF were elevated in 2 breast cancer patients after COVID-19 infection. Next slide. And I'd like to acknowledge the members of the collaborative team that worked together to complete the study. I mentioned many of them throughout the course of the talk, so I won't go into listing the names. And then our funding, including the DaCCoTA grant, which we greatly appreciate and thank for allowing us to participate in this research project that linked basic science with clinical science as well. So at that, I'll take any questions. Thank you.
All right, yep. Open time for questions.
Dr. Hoover, maybe I can ask the question because of curiosity. After last 2 years of vaccination, is there any data indicating that basically vaccination helped to increase the survival of the cancer patients that in the last 2 years also infected by COVID?
Yeah, I don't know all those data. I think they're still kind of figuring some of that out. I do know that the antibody responses, which was nice to see in our pediatric arm, you could see the antibody responses were similar in the vaccine recipients as the both arms. So I think we're still kind of navigating our learning about the virus as we learn about the immune response at the same time. You know, we don't have correlates of protective immunity yet for SARS-CoV-2, so we don't know how high of an antibody level you need to be protective. We do know a recent study came out showing that even low levels of antibodies can, people with low levels of antibodies can be protected against reinfection, but that all depends on what variant is circulating and how that escapes the immune response. So there's a lot of unknowns in our understanding of this. What appears to be, you know, a good sign is that some of these initial populations that we thought would be really at risk have not been as at risk as we thought, more along the lines of what we see with normal respiratory viral infections. And that was something, again, that we didn't know in 2020 when we applied for this, and it would move through this pandemic with everybody else. And we've learned, you know, matches along with other people we're seeing as well. So.
Thanks. I also want to add that regarding to the ApoE, because was kind of interesting finding in the proteomic data, we had a limited number of patients with the diverse type of cancer, breast cancer, colon cancer, ovarian cancer, and skin cancer. And interestingly, when we checked the other sample that we had for ApoE, we didn't see changes that we saw in the breast cancer. It looks that every cancers depend different response to COVID in terms of the whether make it a more poor performance or not basically not answering to the COVID. So this was also interesting finding that the diverse cancer has differently crosstalk with the COVID.
Yes, thank you, Khosrow.
All right, so we got a question in the Q&A. Any differences in the elderly population's antibody responses?
So that's a good question. We were looking more at adult, not elderly. So we didn't have that arm in this. We were adult and then pediatric. And we didn't have good, again, with it being kind of a smaller study with only 15 max per group, we don't have the ability to really separate out age groups in the study arms at this time. I'm sure other groups are looking at some of that now. As we've got a lot more experience with SARS-CoV-2.
All right, last call for questions. All right, well, I want to thank all of our wonderful awardees in this first update session. That was a great set of talks.